Enzymes and metabolism. Metabolism is defined as all biochemical reactions in a cell that are necessary to sustain life. Oftentimes in our common everyday discussions, we use the term metabolism simply to refer to how quickly or how slowly we might metabolize our food and extra calories. But here we want to redefine metabolism to include all biochemical reactions and all life functions. In discussing metabolism, it's intimately related to chemical reactions, and that means we have to discuss atoms and molecules. In liquids and in gases, atoms and molecules frequently collide with one another. These collisions can lead to random bond formation and breakage. With the help of an enzyme, a chemical reaction or collision can be sped up millions of times faster in terms of the rate of bond formation. The actual number of collisions is not increased, but the number of collisions that result in a bond formation are greatly increased with the help of an enzyme. If we were to just boil down what chemical reactions are in terms of biology, we'd only be concerned with primarily two things. If we consider a chemical reaction has occurred, we are saying that a bond is made or broken within or between two molecules. We would also be concerned with whether or not electrons were transferred between one molecule and another. In a chemical reaction, in order for a bond to be made or broken, or electrons to be exchanged, there are two very important characteristics that must be met. Number one, sufficient energy is required to either make or break the bond. Number two, the correct orientation or positioning needs to be addressed when the molecules strike or collide with each other. In this illustration, the reactants on the left are colliding with each other, but not at the correct orientation. Upon collision, they simply strike one another, and both are moved in different directions at different angles at different speeds afterwards. In this kind of scenario, heat may be released, but no chemical bonds were made and no electrons were exchanged. Therefore, in this case, there is no reaction. Many collisions result in no reactions because the angle at which you can approach any molecule is limited very strictly by where those orbitals are positioned. In the second picture, if the molecules strike each other and by luck the orbitals are lined up upon the moment of collision, you can see that it's possible for a new bond to be made or electron to be exchanged. But that would require that there was sufficient energy. So if A struck B but didn't hit it hard enough, then again there would be no reaction. So if both conditions are met, correct speed at moment of collision, which equals enough kinetic energy to make or break the bond, and then if the collision occurs in the right directionality, meaning the molecules are lined up so that the orbitals are in line with one another, then you will produce a product, or you'll have a bond making or breaking event, or electrons exchanged. Let's talk a little bit about how enzymes actually do this. How can they make chemical bonds happen more frequently than just by random collision? The special shape of the protein is the key to how it assists in the reactions. Enzymes are defined as chemical catalysts. The term catalyst refers to any molecule that can speed up a chemical reaction but not be used up or consumed or altered by the reaction in any way. So the reaction does not alter the enzyme, which means the enzyme can continue to react and continue to be used in subsequent reactions, even though the reactants and products are changed. Each enzyme has a very special surface called the active site, where the molecules it assists in the reactions will actually make contact. The term substrate refers to the reactant or reactants that interact with an enzyme. Some enzymes take molecules apart, therefore it has one active site and one substrate. And then when the substrate is torn apart, the bond is broken, then the products are released from that enzyme. Other enzymes have multiple substrates, usually two, and if both substrates enter the active site, they're going to end up being bonded together so that what leaves is a single product. Here in your textbook illustration, we can see a protein that has an active site. So here in the picture, this divot is the active site. The substrate molecule fits into the active site. The active site is very specially shaped so that it only accepts one very specific molecule or substrate. The active site's shape is defined by not just its physical dimensions and actual contour, but also by where specific functional groups are located within the active site. So another molecule of the same physical shape might fit into the active site, but no reaction will occur if the functional groups are not also corresponding. If I follow this with my arrow, you can see that it continues as a single chain. And all of these little dashes in between show different kinds of internal bonds for the primary, secondary, and tertiary structures. The active site on the surface is where all of the side chains and other functional groups are located, and they have to be corresponding specifically to every single functional group on the substrate molecule that it interacts with. So the secret to how an enzyme makes the reactions occur is that it will help position the reactants exactly in the right orientation for the reaction to occur. Enzymes help speed up reactions millions of times faster because every substrate that enters the active site will enter with the right orientation, and when the substrates collide with one another or they collide with the active site itself, then the bond that's going to be made or broken will be positioned in the right position for that to occur. And since these functional groups are attractive, the molecule is actually going to be drawn in faster as it approaches. So the substrate molecules will accelerate, usually to the proper speed for the collision as well. So in the active site, you have proper speed of collision and proper orientation at the moment of collision. We can use just a little semicircle with a divot in it to represent the enzyme. This little rectangular area will represent the active site. Substrate molecules can only enter an active site where their functional groups are complementary. For example, along this surface of the pink substrate, it will only be attracted to functional groups on this side of the enzyme's active site. On the opposite side of the active site, this surface here, it will only correspond to functional groups on this side of this reactant. So when they enter, they will only enter where they're supposed to for the reaction. And if this one is positioned this way and this one's positioned that way, then as they enter, they're going to collide with one another, and the bond will be made where they strike and collide with one another. So the enzyme attracts the reactants into the active site so that the molecules that enter will collide in the right orientation. Upon collision and the bond is made, the products will be released, and then now the active site is reset, so for the next set of substrates to enter. In your textbooks, there's an illustration here that describes how energy changes in reactants to products. So for this specific diagram, we're looking at an energy profile of a exergonic reaction, where the products contain less energy than the reactants. This would be an example of taking a molecule apart, for instance, breaking a bond. In order to break a bond between two atoms, we need to have them collide with each other with sufficient speed and orientation. The amount of wasted energy, then, or the number of attempts or tries necessary to get these bonds to begin breaking or to generate enough heat in the solution to cause others to have enough energy to break the bonds is called the energy of activation. If you think about an example like burning a piece of paper, the paper will not initiate burning until you provide a flame or a spark next to the paper. So in the same way, in a chemical solution, in order for a bond to actually break, it will have to collide a certain number of times wastefully, not being able to break that bond before ultimately it results in that bond breakage. What happens when there is no enzyme present is that the amount of collisions is going to be relatively high before you actually get the bond to break, if we're doing this in a random collision fashion. But you see what happens is the yellow line represents a lowered energy of activation when an enzyme is present. So fewer of the collisions will be wasted, less heat will be produced, and the same number of bonds will be broken in the same amount of time with the help of the enzyme.